All right. Good morning, everyone. My name is Bernd Willems. I am the Regional Application Specialist for Twist Biosciences Synthetic Biology portfolio. I am delighted to welcome you all to the Twist webinar with the title, Using Next Generation CRISPR Screens to Advance Our Understanding of T-Cell Biology. This webinar consists of two parts. In the first part, our dear colleague, Dr. Julian Jude, will set the stage by giving a thorough introduction to CRISPR screening and will highlight the significance of Twist oligo pools as a valuable tool for this application. Next, our valued guest speaker, Dr. Hongling, will tell us his story about he and his team have been using in vivo CRISPR screens to reveal novel regulators of T-cell fate decision. But before introducing our speakers, just a few housekeeping items. All lines will be muted during the webinar. We will answer all questions at the end of the presentation. If you have any questions, please submit them in the Q&A box found at the bottom of the screen. Following the presentation, we will invite you to take 1 minute to do a very brief survey about your webinar experience. We would really appreciate your feedback. Now, I would like to introduce our first speaker, our colleague, Dr. Julian Jude. Dr. Julian Jude completed his PhD at Cancer Research UK Manchester Institute, developing an arrayed shRNA screening platform. He then moved on to do his postdoc at Prof. Johannes Zuber's lab at IMP in Vienna, Austria, where he developed functional genomics tools. He performed over 70 genome-wide shRNA and CRISPR screens, probing for drug synergy, synthetic lethality, and drug targets. He collaborated closely with Dr. Ulrich Elling at IMBA to develop the VBC score sgRNA design algorithm. Dr. Jude then became a team leader at Alien Biotechnology to build a crop-seq screening platform, merging single-cell CRISPR screens with single-cell RNA-seq, before changing gears to join Twist Bioscience. Julian, thanks for being here today. The floor is yours.
Hello, everyone. My name is Julian Jude. I'm a scientific development manager here at Twist Bioscience, and I highly appreciate it that you take the time and dial in to learn about how Twist is fueling the CRISPR revolution with our limitless synthetic DNA. So this presentation is going to be split in two parts. The first part is about how Twist is synthesizing these next generation synthetic DNA tools, and the second part is how the clients, the scientists in the field, are utilizing the synthetic DNA to drive the CRISPR revolution forward. So at Twist, we believe that synthetic DNA has the potential to solve many critical challenges that face humanity today. Critical challenges such as finding ways to produce materials and chemicals in a sustainable manner, also to ensure our food security, diagnosing and treating human disease, and last but not least, also managing our ever-growing need to store data. And not only are we able now to write the code of life using synthetic DNA, but we are also able to engineer it at ease. And this is done using a family of RNA-guided bacterial nucleases with its most famous member, Cas9. And this CRISPR revolution enables scientists to probe and engineer the genome at will, getting us faster to viable solutions to these challenges that I've mentioned. So how is Twist able to accelerate this CRISPR revolution? Well, we have created a transformative platform for DNA synthesis based on DNA writing using silicon as a substrate that allows researchers to move beyond what was previously possible. It's not just a massive increase in scale that we are able to drive this platform, but a big improvement in sustainability as we move away from hundreds of plastic plates to synthesize DNA to a silicon chip that is able to synthesize 10,000 times more oligos on the same footprint as a 96-well plate, generating only 96 oligos. And this makes synthetic DNA and its associated experiments scalable like never before. But something to stress is that the oligos synthesized on a twist silicon chip are extremely uniform and have an incredible low error rate of 1 to 2,000 base pairs only. And this means we have a raw material, synthetic DNA raw material, that allows us to build extraordinary synthetic tools for our clients. And it's this DNA engine that underpins all of our products and services. And it's how we're supporting our clients and collaborators with a huge variety of applications. And today I want to talk about these products, especially the twist oligopools, and how they are supporting scientists in accelerating the CRISPR revolution. So now let's switch gears a bit. I'm going to talk about CRISPR for a second. So as this audience knows way too well, CRISPR-Cas9 is a functional genomics power tool, and it's very basic. You have the Cas9 nuclease, bacterial nuclease, which has been domesticated to work in any organism that you can think of nowadays. But ultimately, the Cas9 is loaded by the sgRNA. So this is the guidance mechanism for the Cas9 to find a specific spot complementary to the sgRNA into the genome, and there will cause a double-strand break. And this break can be repaired in 2 different ways. The first way is a non-homologous end-joining pathway, which is a bit error-prone, leading to insertions in the lesions in the double-strand break. And therefore, if this happens within an exon, we have an out-of-frame mutation called eventually turning into a gene knockout. The more precise mechanism, the homology-directed repair mechanism, allows you to actually supplement this reaction using a DNA template. And this template is then inserted into the double-strand break in a very precise manner. And this allows you now to do something called the gene knock-in. So you can even tag your gene of interest in the genome with a fluorescent marker, for example, if this template DNA encodes for GFP. And this is happening rapidly and at ease. So it's very easy to design these guides and engineer the genome nowadays. And because it's so easy, scientists have now went on to ask the question, how important are the genes in the entire genome for a specific phenotype? So they're building CRISPR screens. And essentially what happens is that you start off by designing a library, meaning you design all the sgRNAs to target all the genes in the genome. These sgRNAs are then sent to Twist, for example, where we synthesize them as DNA oligos. These are then being cloned into your plasmid of interest to express the sgRNA. And normally what happens is that you have the cloned oligopool, so the plasmid pool, converted into viral particles, which are then used to infect target cells that express Cas9, where then the guide is taking Cas9 and knock out the single gene in this genome. And you do this for every single gene that you have in the genome. And you can ask the question, how many of my guides are present at the beginning of the experiment and how many guides are present at the end of the experiment? In this case, it would active pressure as proliferation. So ultimately, the data you will get out is a gene essential for the cell proliferation or not? If the sgRNAs targeting a specific gene are present at the beginning of the experiment, but not at the end of the experiment, it means that if you knock out this gene, the cell cannot survive or cannot proliferate and will drop out. And this can be done now at scale. So we can assign genotype to phenotype at a scale in an unbiased manner that has never been possible before. And it's paramount to perform CRISPR screens of high quality reagents to be confident when you call these hits at the end of the experiment. So twist oligopools fueled CRISPR screens have the competitive edge that they have a high signal to noise ratio, which makes it much easier to call a hit. And therefore, it's less validation work required downstream, and therefore it saves a lot of time and money. And this is because of the very tight distribution of our oligopools that come off the chip that feed into this cloning pipeline. And ultimately, you have a very high signal to noise ratio. And it looks like this. It's very easy if you have a very even background at the beginning of your experiment to call these hits because they will be standing out from the data. But if you have an uneven uniformity where some of the guides will be overrepresented already at the beginning of the experiment, there's a low signal to noise ratio, and therefore everything looks a bit like a hit. And therefore you have to pick more potential hits that might be false positives, and these can only be discovered if you then perform a lot of validation experiments downstream. For this 2019 study, scientists at the Sanger and Cambridge UK have relied on the high-quality twist oligopools to build their genome-wide CRISPR library, which was eventually screened in over 324 human cancer cell lines. And these represented 30 different types of cancer. And the resulting data from this massive screening effort was the foundation of a framework to prioritize candidates for cancer therapeutics. And this is a type of genome-wide knockout screens that I call single-guide CRISPR library screens, because they only have a single guide on an oligo that's being cloned into the expression vector. So you will only knock out one gene in a single cell. But we at Twist are always revolutionizing the field. In this case, we actually are able to synthesize oligos up to 300 nucleotides in length with high quality. And this really opens a completely new area of functional genomic screens that can be performed. Because of this long nucleotide that can be synthesized, we can move away from single guides to actually dual and even triple or quadruple guide libraries, depending on what kind of enzyme you're working with. So in this case, we have 100 base pairs, which is an sgRNA and a tracer. And then the same one is sgRNA tracer and a tRNA, another sgRNA, which are all encoded on the same oligo. And this means that you can deliver into the genome of a single cell a virus that expresses two guides at the same time, which are with matched pairs. This means that high-quality oligos are able to perform, enabling the next generation of precision CRISPR screens that were previously not possible. So the question, what can you do with such a matched system, right? Where you have the first guide and the second guide on the same plasmid together designed. The first thing is, of course, that you can delete now genetic elements like long non-coding RNAs. So these long non-coding RNAs in the cell are not expressed just like genes. That means that there is no out-of-frame mutation that you could generate with a single guide. You need to really cut these out of the genome, like with two scissors. And this can only be done by having two guides in the cell together on the same plasmid. So they're expressed together and you have the cutting effect in the same time. You could also imagine increasing the loss of function rate, right? If you have a single guide in the cell to cut the exon of your gene of interest, there is a chance that it will generate an in-frame mutation and then you don't have a loss of function, right? It will still express the gene at the end of the day and you will not see a phenotype. But to increase the loss of function rates, you can have two guides targeting the same gene at different parts of the exon and therefore increase the rate of a loss of function phenotype and therefore knockout efficacy. If you're working with CRISPR activation or inhibition, so CRISPR-I and CRISPR-A, you can have a tiling effect by having two guides targeting the transcriptional start site of your gene of interest and therefore have enhanced activation activity because there are two entities of Cas9 targeted in front of the transcriptional start site. But last but not least, what I really like about this technology is that you can now systematically probe defined gene pairs. And what does this mean? So it means that you can have a combinatorial screen where you knock out one gene as a pair with all the other genes that you are interested in. And one way or one outcome of these screens are that you find synthetic lethal partners. That means in a normal scenario where if you knock out one gene, there will be no phenotype. And if you knock out the other gene, there will be no phenotype. But if you knock out both at the same time, because they have compensating functions, if you knock out both at the same time, there will be a phenotype. Of course, it's very interesting to find new drug targets for cancer, because we know that cancer genomes are mutated. So if you imagine you sequence the cancer genome and you find these mutations, that could be one set of guides, so you target all of the genes that are being mutated, and the second set of guides are all the other, for example, FDA-approved drug targets, and you try to find a match there. And the beauty here is that, of course, all the normal cells in your body will not have this second set of mutations. Therefore, it's much easier to find a treatment that will specifically target the cancer. Now, moving forward, there's, of course, the 300 nucleotides again, utilized in a completely different way. And one new technology that comes from David Lee's lab is also known as search and replace technology. And this is a fusion between a modified Cas9, as you can see here, with a reverse transcriptase, the top part here. And this technology is called prime editing. And it's also known as search and replace, as I mentioned, because what you can do now is encode into the prime editor pegRNA. This is this really long pegRNA as depicted here. You can encode into the end the mutation that you would like to have introduced into the genome. So how does it work? You have the normal sgRNA in the tracer, which will guide the Cas9 to the specific loci in the genome. And instead of just causing a cut, it will actually have this long tail here, which is reverse transcribed by the reverse transcriptase into single-stranded DNA. And then this part in green here will actually align to the section in the genome which is SNC'd, and then it will incorporate this little green part here into the genome because it's search and replace, right? So it will introduce the mutation that you encode into the genome. And this is, of course, only possible to do such an experiment and scale if you have high quality oligos that go very long in their length, up to 300 nucleotides. Because you don't want to introduce any sort of synthesis errors that may be caused during the synthesis of these oligo pools or low quality oligo pools. Because you eventually will not sequence the genome for the outcome, you will only know what kind of oligos have been used by sequencing the plasmid pool that comes from the cloning reaction. So it means high quality, long oligonucleotides are enabling prime editing at a scale which has not been done before, just because you're confident that the mutations that you encode into the part of the pegRNA will actually be incorporated into the genome, and it's not the mutations from a synthesis error. Now, of course, prime editing is a very early technology, and people or scientists are always trying to improve these. And nowadays, there's always this buzzword about deep learning and AI. And of course, it has made all its way also to the prime editing community. And this is a very beautiful way to use these long 300 nucleotide long oligos from Twist Bioscience, because what you can do and encode on these 300 nucleotides is multiple elements that help aiding to identify novel algorithms to design these specifically optimized pegRNA guides. So one of these experiments to train a model to build these new prime editing design rules is by that. You have a lentivirus, which encodes a pegRNA, a tracer, then also the rest of the pegRNA, the PBS and the reverse transcriptase site. But on top of it, you also have a target site and a PAM sequence. That means if you infect a cell that expresses a prime editor with this oligo containing lentivirus, the prime editor will take the sgRNA and actually jump on the same part of the oligo, the same part of the virion that is integrated into the genome, and cause an edit. And now this makes it much easier to identify this edit because you know the surrounding sequences, and you can use a universal primer to amplify this part out, and then NGS sequence and identify the edit that came out of this. This data is then fed into the deep neural network, which then allows you to design new design rules because you can predict what the editing outcomes is based on this data set that is generated through this. And this can be done at scale. Because if you would do this just for a single pegRNA, it's easy because you know where in the genome it's landed, and you would amplify this out. But to do this in a scale where you use thousands of these target regions, you would need to also synthesize 1,000 different primers and amplifiers out from the genome. But if you use this technology here, the high quality long nucleotides from Twist Bioscience, you are able to prime on the constant regions on these integrated viruses, and then therefore assay thousands of different genes at ease. The take-home message is really that with this longer high-quality oligo pools, you are able to generate data sets for deep learning that enable an optimized pegRNA design algorithm. And of course, this approach, this high-throughput approach to design new guide algorithms has been also utilized for Cas9. But because the Cas9 guide and tracer is much shorter than the pegRNA, you can even extend this experiment and perform something completely novel, which is not only assaying the on-target activity, but also the off-target activity. And there's these papers on the side here, which if you are interested in this, you can dive deeper, but I will just try to give a quick overview on this part of the presentation here. So similar to the pegRNA, you can have a lentivirus that expresses the sgRNA tracer. The first on target, so there's 100% matching to the sgRNA target site, but you also can now introduce, because you have enough space on the 300 nucleotides, to have a second target space, which could have a few mutations away from the wild type, from the matching sgRNA. And again, you can do this at scale. And if the Cas9 edits only the first target, which has 100% match to the sgRNA, but not to the off-target side, it means that the design of this sgRNA is very efficient to have an on-target activity and almost no off-target activity. Again, you can amplify this out, sequence it, and then have a deep neural network identify the design algorithm that allows you to have these optimized on-target sgRNA designs. Now, of course, if you have an editing effect where you have a target, the first target, which is 100% match of the sgRNA to be edited, but also the off-target sequence, the bait for the Cas9 to cut there. If it cuts there as well, then you know that this design of the sgRNA has a flaw, because not only will it cause on-target activity, but also off-target activity. And again, these sort of experiments where you have an NGS output at the end of the experiment, if there's any sort of data sets that are then generated that go into deep neural network, you would like to have as a minimum of any sort of synthesis errors, because the training set will then train the deep learning algorithm on the synthesis errors on top of the editing outcomes that come from the Cas9. And this is not something you want. You want to have a pure data set that only goes into a deep learning neural network that has the mutations from the editing outcome and not any sort of synthesis errors or biases. And this really allows us, with the long, high-quality oligos that are being generated from the Twist Silicon platform, to build data sets for deep learning. And this allows us to build a new type of sgRNA design algorithm that has an increased on-target activity versus off-target activity. And I say, and with this slide, I would also like to conclude. So I hope I could convince you that the CRISPR revolution is fueled by Twist's long, high-quality oligos that enable scientists to move beyond single guide CRISPR libraries and experiments to venture into a more multiplex and even prime editing screens at scale. And of course, the high uniformity of Twist board allows for confident hit calling and high signal-to-noise ratios, which means that it's much easier to identify the hits, and therefore you have to do less downstream validation experiments. And ultimately, the data sets that are generated are clean enough to go into AI-driven guide designs to build the next generation of CRISPR sgRNA designs, as well as prime editing designs. So if you want to find out more, last but not least, I invite you all to browse through our webinars hosted on the Twist webpage, featuring some of the most exceptional CRISPR screeners, testing their work from single-cell combinatorial screens all the way to synthetic lethal screens, and even novel sgRNA designs that have a more on-target activity. So thank you very much. And if you have any questions, please send them over. Thank you.
All right. Thank you very much, Julian. That was very thorough indeed. And so now it gives me great pleasure to introduce today's guest speaker, Dr. Hongling. Dr. Huang completed his PhD at Shanghai Institute of Biochemistry and Cell Biology in China in 2013. Following his PhD training, he pursued his first round of postdoctoral training in the laboratory of Dr. Peter Camelier at VIB-TU Leuven in Belgium from 2014 to 2016. In 2017, he joined the Department of Immunology at St. Jude Children's Hospital in Memphis, Tennessee, in the lab of Dr. Hongbo Chi as a postdoc. Early this year, he started his independent research as principal investigator at Xiamen University of China. Well, congratulations. Dr. Hongling's research interest lies in the immune signaling underlying the differentiation and function of T cells, a central cell type in adaptive immunity and cancer immunotherapy. He applies systems biology approaches such as single-cell RNA-seq, CRISPR screening, and the integrative network analysis to reconstruct molecular hubs for T-cell fate decision in tumor infection and autoimmune disease, as well as in healthy condition. His work could shed light on understanding fundamental mechanisms of immune regulation and pave the way for better immunotherapy. So without further ado, let us welcome Dr. Huang to start his presentation. Dr. Huang, thank you so much for being here with us today. The floor is yours.
Okay, thank you. So thanks a lot for the kind introduction. So also thank Twist to give me this opportunity to present our work, and also thank my previous supervisor Hong Chi for the recommendation to attend this webinar. So today I'm going to share how we use the in vivo CRISPR screens to dissect the T-cell fate decision. So please allow me first to introduce some background. Actually, our lab has long-term interest to study so-called bidirectional interaction between T-cell and cellular metabolism. So basically, we are interested in how T-cells engage cellular metabolism to pursue its function, and also how the cellular metabolism can interact with to direct the T-cell functions. So back to more than 10 years ago, so we also used, in our lab, we also used a lot of traditional conditional knockout strategy. Basically, we deletion one gene and study the role of this gene in T-cell biology. And then we recently realized that the cellular interaction network is extremely comprehensive. So it's impossible that to deletion every metabolic gene and then study the function of this gene in T-cell biology. And also there are some redundancy of cellular metabolism pathways. Therefore, in recent years, we try to use some kind of systems immunology approaches. For example, we generate some algorithm and use bioinformatics tools to study the to predict some important cellular pathways in the T-cell biology. And also we use some systems proteomics approaches to find out the key protein that may change during the T-cell activation and also during the T-cell development. But recent years, we also used another approach we called in vivo CRISPR screening. And this is also the focus of this talk today. So we use this approach, also published several papers recent years. And then I will just use this the paper that we published in Nature, and as an example how we use this in vivo CRISPR approach to study T-cell biology in vivo. So basically we generate a focus library that targeting metabolic genes, and then we packaging virus, and then we use the virus to transduce antigen-specific cells, and then we transfer those antigen-specific cells to the tumor-bearing mice. Because those tumor antigen-specific cells, they are able to recognize the tumor in the host, and they once they recognize and being activated, they can start to proliferation and undergo differentiation. And after several days, after the T-cell proliferation, so when we harvest the tumor infiltrating T cells, and then we did guide RNA sequencing, and then which allows us to compare the guide RNA enrichment in the tumor infiltrating T cells and compared to the pre-transferred input. So in this assay, actually we use a very, very simple result, which is the T-cell accumulation as a result, which allowed us to find out regulators 1 actually is a very key negative genes that control T-cell accumulation in tumor microenvironment. And also from the mechanism study, so then we found that regulators 1 deficient T cells, actually they were reprogrammed to a memory-like T cell fate, and those reprogram can enhance the T cell function to kill tumor cells, which can be benefited to control the tumor. So this study not only showing that it's feasible to use the in vivo CRISPR screening approach to study the T cell function in vivo, but also give us a hint that so the T cell function and the T cell differentiation status is highly closely related to each other. So due to this, then we had an idea, why don't we use the also use the in vivo CRISPR screening approach to study the T cell fate decision in vivo. So because regulators 1 knockout cells actually gain some kind of memory T cell features. So then we had a thought, can we use in vivo CRISPR screening approach to study the memory T cell development? And here I'm showing you just a typical curve of antigen-specific cells. We all know that once the antigen-specific cells recognize the antigen from virus or bacteria, they undergo a rapid expansion phase and undergo effect cell differentiation. And once the bacteria or virus was cleaned, and those cells undergo rapid contraction phase followed by a stable memory cell formation. And those antigen-specific memory cells, they are able to activate it and become second effector cells once the virus or bacteria is challenged again. So there are different models how the memory cell is formed in vivo. So here I'm just show you one a very popular model. So if we only look at the cells in the expansion phase or effect phase, there are two types of cells. So one called terminal effect cells, or in shortly called T cells. Another type of cells called memory precursor cells, or MP cells. Compared to MP cells, T cells are likely become cell death once the antigen was cleaned. And on the other hand, MP cells can serve as the precursor for the memory cells, which can persist much longer compared to the terminal effect cells. Because our initiative was to identify any gene regulator can promote memory cell generation. So then we had an idea that just the blockage of the terminal effect cell differentiation, and those cells may undergo the memory precursor cell fate and generate more and more memory cells. To this end, actually, then we designed this kind of in vivo CRISPR screening system. So basically, we synthesize the oligo from twist, and then we use CRISPR assembling to generate a focused library. And then again, Then we again just pack the virus, and then we use this virus to transduce the antigen-specific cells after several days of in vitro culture to allow the gene editing to occur, and then we sort out those transduced cells and transfer those cells to the naive host, followed by the LCMV infection. This because this antigen-specific cells will recognize the antigen in the virus, and those cells will undergo rapid proliferation and differentiation. So at the 7.5 time point, which is the point of the effector phase, so we then sorted out the MP cells, memory precursor cells, and the T cells or terminal effector cells based on the surface marker of these populations. And then again, we did the DNA isolation followed by the deeper sequencing. then compared to the import, so then we are able to find out which gene is selectively depleted in the LMP cells or T cells, or which gene is selectively enriched in these two populations. And then follow this by informatic analysis, and actually we found out a lot of positive controls which have been shown in the fatal decision of the LMP and the T cells was found out in our system, means that the system was working. And then we also found another gene called the PLF51, which is an enzyme that can force used to the fucose as a function as a fucose transferase. And then we validated this gene by a system we called a dual color transfer system. In brief, so in this system, we label the non-targeting controls with GLP, and then the label with knockout cells with amycin, and then we mix these two cells together at a 1:1 ratio and transfer those cells into the live host, followed by the virus infection again. And then we monitor those cells at the different time points post-infection. Basically, there are two main time points we were interested in. First time point was the effector phase. Again, means that the day 7.5 post-infection, and also the memory phase, which is more than 30 days post-infection. And then at the effector phase, we can clearly see that once PLF51 was knocked out, so we can see those effector cells proliferate, accumulated in the spleen much more compared to the non-target control. And also, we again use just calculate the number of the effector cells. We can also find out PLF51 deficient cells that can greatly enhance the effector cell accumulation in all the organs we have examined so far. And then so how about the memory cell response? So then we again also calculate the number of memory cells at the 30 days post-infection. Again, we saw that knockout PLF51 can greatly enhance the T cell accumulation in all the organs. And how about the function of those memory cells? We used the two types of experiment to examine the function of those memory cells. One we called in vivo killing assay. In this assay, we can clearly see that the mice that bearing the memory cell of PLF51 memory, knockout memory cells can clean those target cells much rapidly compared to the non-targeting control mice. And another assay, we also used the tumor challenging assay to challenge those mice that had been received the PLF51 deficient cells. Again, we found that PLF51 deficient can greatly enhance the tumor control and also prolong the tumor the survival of those mice. So in summary, so actually in this part, we just established an in vivo pooled CRISPR screening to study the fate decision of the effector cells. And also we found that we identified one candidate gene called PLF51, which can improve its deficiency can improve both the effector cell and memory cell response. I apologize that I am not able to show a lot of data, but here I just show you one conclusion that there are two key features of PLF51 deficient cells. One is that it can inhibit the terminal effector cell differentiation, and another key feature is that it can enhance cellularity of effector cells. Because these two features actually is not commonly you can see from the other knockout. So based on this finding, then we had the idea, so why PLF51 have some kind of unique features. So due to this, then we had the idea that maybe PLF51 deficient cells can alter the cellular status. So to this, so then to answer this question, then we use a single cell RNA-seq to study to understand the cellular differentiation status of PLF51 deficient cells. Here we use I present a UMAP plot, and you can see in black cells, they are all non-target control cells, and the red cells, they are PLF1 deficient cells. If you overlay these two plots together, you can clearly see that PLF1 T-sequencing cells actually they are not able to differentiate to some certain status of the cells on the left corner. So then we just basically we just analyze the surface molecule of those cells. We can see those cells, they highly express of KRAS1, low expression for CXCR3, and low expression for the interleukin-7 receptor. So with this surface marker, then we again just to rename of the different status of cells. We call one cells is T prime cells. This is also a kind of terminal effect cells, but it's different from the terminal effect cells I mentioned before. So then we call this state of cells as a T prime cell. We saw a lot of the intermediate cells, and then we also saw the memory precursor cells. Then we used the flow cytometry to validate our finding. Again, we found that a loss of PLF81 can significantly reduce the T prime cell differentiation. On the other hand, it can promote the intermediate cell accumulation. So as an immunologist, we always want to know, since we use the different marker combination to find out the different status of the T cells, so we want to know how about in the Y-type context. Can those cells can be also found in the Y-type cells? So indeed, we use the identified markers. Then we also able to sort out the different population of cells from the Y-type context. And then we did a microarray, and also we found that those cells also have very different transcription heterogeneity. And so means that so not only in the knockout context, in the Y-type of context, we also can see these three effector subsets. And also we do the GSEA and also some some more analysis, and then we can see those three subsets really exist in the Y-type context, and those cells display the transcriptional heterogeneity. And then here I just again summarize the data I showed you before, and basically we by combination of the CRISPR screen and single cell RNA-seq, so we are able to identify some three different status of effect cells, and then we found that the key role of PLF81 here is just control the differentiation from the intermediate state of cells to the T prime cells. And then the next question become very clear. So what's the mechanism? How does PLF81 control the intermediate cell to the terminal effect cell differentiation? To answer this question, we first need to know what's the role of PLF81 from the literature. We know that PLF81 is a protein O-fucose transferase. So, which means that PLF81 is an enzyme that can transfer the fucose from GTP fucose to the target protein. So, according to the literature, there are more than 100 proteins that can be potentially as a target for the PLF81, including the Notch receptors. So then we first want to know, so does the fucose availability is essential for the T prime differentiation? To answer this question, first we generate a mutant form of PLF81. This mutant form of PLF81 is not, it can lose the function of the fucose transferase activity. So we found that if you overexpression the Y-type form of PLF81, this form can fully restore the PLF81 deficiency induced the T prime cell differentiation. But if you overexpression the PLF81 mutant form, so which are not able to restore the PLF81 deficiency induced T prime cell differentiation defects. So which means that PLF81 relies on its fucose transferase activity to promote the T prime cell differentiation. And again, then we directly examine if the GTP fucose is essential for the differentiation of T prime cells. So GTP filters in the cells, GTP filters can be synthesized from two independent pathways. One is called the de novo pathways, which is mediated by a key enzyme called GMTS, and another pathway called the salvage pathways, which is mediated by another enzyme called FCSK. So we generate a double mutant for the GMDS and FCSK. And we clearly can see that if you generate this mutant, the cellular GTP filters level can be significantly reduced. So in this context, when we check the T prime cell differentiation, again, we saw that double mutants can fully blockade of the T prime cell differentiation. And again, we know there are two features of PLF1 deficient cells. Those cells can also increase the number of the effector cells. So again, we found that when you double deletion of GMDS and FCSK, so those cells, effector cells, also have higher frequency as a number level. So all of this evidence is showing that PLF1 relied on the GTP filters and also filters availability to promote the T prime cell differentiation. So the next question is that what's the downstream event of the PLF1? So to answer this question, we basically just did a simple microarray assay, and then we again do a gene set enrichment analysis using the microarray dataset, and then we found that PLF1 deficient cells can greatly reduce the notch signaling. Actually, notch signaling was the only significant reduced gene in the hallmark collection. So it means that maybe PLF1 control notch signaling. And to further validate this hypothesis, so then we directly examine the NICD expression, which is the notch intracellular domain. And then we can see loss of PLF1 can greatly impair the NICD protein level. So again, means that PLF1 control notch activation. So to further validate that PLF1 is function as a notch activated to promote T prime differentiation. So then we did a rescue experiment. So in this experiment, we knock out PLF1 and then in the knockout context, we overexpression NICD. So if PLF1 slew the downstream NICD to promote T prime differentiation, so then we should expect NICD should fully restore the function of PLF1 in this context. Actually, this is was exactly what we saw from our microarray assay. You can see that loss of PLF1 can change the transcriptome compared to the Y-type control. And also, if you overexpression of NICD, so the transcription induced by the NICD expression is completely different from the PLF1 deficiency. And if you combine these two together, then we found that NICD overexpression can fully restore the transcription level activity induced by the PLF1 deficiency. And then we use the flow cytometry to validate our finding and then examine the T prime cell differentiation. Again, we found that loss of PLF1 can greatly reduce the T prime cell differentiation, but if you overexpression NICD, then it can fully restore the defects induced by PLF1 deficiency. And also, we also calculate the number of effector cells. Again, we saw that PLF1 can increase the effector cell number, and then once you overexpression NICD, it can greatly reduce the number of effector cells. So basically, from this study, actually, we started our project from in vivo CRISPR screening, and then we designed this system to study the fatal decision of memory precursor cells and the terminal effector cells, and then by combination of the single cell RNA-seq, we are able to find some heterogeneity of the effector cells. Basically, we found three subsets of effector cells. And then we realized that the PL51 and the downstream notch is very essential to control the intermediate cells to terminal effects of differentiation. And then if you are able to blockade the PL51 induce the terminal effects of differentiation, we are able to improve the anti-tumor immunity and also generate more memory cells, which also allow us to increase the in vivo killing capacity of those memory cells. With all here, so I would like to make an acknowledgement of everyone get involved in this project. Also, first is my supervisor, Hongbo Qi. Without his guidance and support, we are not able to do this work. And also, there are a lot of lab members in Chile help us to finalize this study. And also, there are some fantastic core facility in Senju help us to finalize this study. And also, thanks all for your attention, and I am ready to take any question. Thanks a lot.
And that brings us to the end of our webinar, and I'd like to thank you all for joining us today. If you'd like to learn more about how Twist products can help you in your research, please visit twistbioscience.com or reach out to us directly or to our regional distribution partners. Thank you again, and enjoy the rest of your day.